pedagogical practice, which means I thought about my teaching, these words like reflective pedagogical practice. And I want to mix things up because I'm boring the snot out of myself, and I imagine that that's doing to some of you, at least those who are not dedicated virus lovers. And there are some of you up there. So what I'm going to do is add a component to the course, and I'm going to make it real. I just can't imagine filling up all of class time with what's in the chapters in the text, and I've thought about how to supplement it. And so here's how we're going to do it. Every Wednesday, at least for the next few weeks, we'll see how it goes. I'm going to spend the last half hour or so of class discussing a paper from the literature. That paper from the literature is going to be one of the preceding week's TWIV podcast papers about which they discuss. So if you're doing the Listen and Reflect the podcast, you'll hear the scientist wants to get done talking about the weather. Good half hour, 45 minutes in. Talk about the latest in virology papers that have been published in recent days. So the reason I'm doing it the previous week is not only to give you a chance to listen to the podcast and do the reflection, but we don't have subscription access to a lot of these journal articles, so I have to get them through interlibrary loan, which can take a couple of days. So this, and just to make it real, I'm going to ask questions about this on the exam, so I encourage you to read the paper, understand as best you can, bring it to class, and we'll have a discussion of it, not only getting you into what it is they're doing, but the formation of the hypotheses, how they're tested, what the data mean, do they do the appropriate controls, are there conclusions, reasonable given the data. So the first one, and these will be put up on D2L under weekly paper discussions. So the first one is from last week's TWIV about cauliflower mosaic virus. So hopefully we'll throw a little spice into our courses. So the ones for this week and then the next week are both up online because the TWIV podcast is on this week. One other note, I think I have replied to everyone. There's been a dramatic expansion in the number of people doing by the way, those who are not in the know, TWIV is This Week in Virology, the hottest, coolest, hippest virology podcast on the web. First week, one person did it. Second week, 5 people did it. I'm not exactly sure how many people did it this week. I encourage all of you to do it. Don't email the reflection to me about 7 people did that this week. Put that up on Turnitin, and I'll grade it on Turnitin, and you can look at what I say and what Turnitin says about the grammar. But anyway, a chance for you to do some writing, to get some cutting edge virology, and now to get a leg up on in-class discussions. Okay, so today we're talking about virus attachment and entry. And your textbook comes up with the worst mnemonic device I've ever seen in my life, which is @gay. It's supposed to remind you of the steps for viral replication, or more informally, the life cycle of the virus. Conveniently color-coded these for you today. We're going to talk about attachment and entry. Wednesday, transcription, translation. That may spill over a bit into next Monday, genome replication, then assembly and exit. So the next section of the course is going to be the generalized life cycle of viruses. But do note, not all viruses do all of these, and some do other things. So this is a general, not quite flow chart, or list of things to do for a virus on causing an infection. I like to think of, rather than the mnemonic device, as @gay. I like to think of my younger days and what is it I need to have a successful one-night stand. You have to go to the bar, find your host. Never mind. But there is some nakedness. You do it right. Okay, attachment occurs between a virus and its host, mediated by a viral cell surface protein and a cell surface protein of the host. So we're going to actually go away from the text and talk about some intermolecular interactions in hopes of having you understand this a little better on a molecular basis. So, let me also start out. I don't give a rat's patootie that you guys memorize the minutia of things like table 5.1. If you look in your text, there's particular virus, particular name of a coat protein like VP3, and the cell surface protein with which it interacts. That's not the sort of stuff I would expect. I would even hope that you would. I hope your life would be better. I wouldn't expect you to remember it 2 years from now. Life is good enough. Or you're either, well, if you're a virologist, perhaps you do. But if you're off in medical school, you should know the generalities and the way things work. But I don't care that you memorize the stuff. I'm not going to ask you match the virus protein with the cell surface protein, except insofar as we talk about it a little bit more, which we're not going to do. So I'm going to take as an example CD4, which is a host protein on helper T cells. GP120, which stands for glycoprotein 120. This is the HIV surface protein that binds to CD4, or the host. And the natural binding partner of CD4 is MHC. There are actually multiple ones, but the one I'll use as examples here are MHC2, major histocompatibility complex 2. If you want to know a lot more about the biology of this, you're going to have to take an immunology course. But MHC2 binds to CD4 in the response of the immune response. So CD4 is the cell surface protein on helper T cells. So it's essentially helping to say, hey, there's something going on here, we need to have an immune response. For virology purposes, know that HIV attachment to the cell is mediated by an interaction between GP120 and CD4, where MHC2 is the normal biological binding partner of CD4. Which brings me to the vocabulary here. When we're talking about attachment events, binding events, when we're talking about attachment, we're talking about binding. We're talking about an interaction between two or more molecules.
It's going to keep going.
That's not the cops coming from this. Just in case it is. We're talking about the protein we're interested in, the ligand, and its receptor. So binding is the interaction between a ligand and its receptor. And they're, of course, interchangeable. So GP120 is a receptor for CD4. CD4 is a receptor for GP120. It's something that binds to something else. Going back to our one-night stand, at gay rather, we have various flavors for enveloped versus naked viruses. So our virus particle is free in solution. It must attach to the host. That's mediated by a receptor interaction. So this is not, when we think about receptors, my personal favorite being the cannabinoid receptor, which is the G protein coupled receptor in U that binds to the active ingredient in marijuana smoke, THC. That receptor does not exist so you can get high. There's a natural function for the cannabinoid receptor, which is neurotransmitter. Actually, there are a number of natural functions for the cannabinoid receptor. It just so happens that I've heard people who smoke marijuana do so to get THC into their systems, at which point it binds the cannabinoid receptor. The name cannabinoid receptor, because that's how it was discovered. THC is the ligand for the cannabinoid receptor. The cannabinoid receptor is the ligand for THC. None of it has a real biological function. We didn't evolve to interact with THC. Well, not most of us didn't. There's no evolutionary pressure for interaction with THC. It's a happy coincidence. Viruses, of course, have evolved to interact with host protein receptors, but the receptors aren't there for the purposes of interacting with the virus. So, in any event, we have a biomolecular interaction event. That's attachment. Your textbook makes a hugely incorrect, and we'll dissect this, statement on several occasions that first, we have weak, reversible interactions, and then with interactions with multiple receptors. This makes sense because, of course, the virus this was icosahedral, so it has the same proteins displayed at different points. It makes sense that they could bind to different receptors in the cell surface. We have multiple interactions that result in irreversible attachment. None of that's not chemically speaking true, it's practically speaking true. At which point we have some sort of entry into the cell. The example here is in clathrin-mediated endocytosis. So clathrin is a protein that helps organize the liposome. We'll see more of this later in the course, into this invagination, which is then pinched off into a vesicle. So there's a cell-derived lipid vesicle surrounding the virus particle, and then the clathrin will dissociate once it's endocytosed, and then through some poorly understood yet obviously event that actually occurs, we have release of the virus. Sorry, that's the dissociation of clathrin. We then have the virus in liposome, which is endosome since it's inside the cell, and somehow that breaks apart and the virus escapes and begins its replication cycle this time. Steve. Kyle.
Travis.
Travis, okay.
Is it safe to assume that this clathrin-coated vesicle is in the same icosahedron shape?
So the clathrin-coated vesicle is not.
Okay.
Right, so the biophysics of a lipid vesicle are going to be spherical.
Okay.
And that goes to the nature of the structure of lipid. If I remember, we've got a double bond in one of the lipids, that's going to form macroscopically, and by macroscopically I mean at the vesicle level, a spherical surface. You can see the virus, which does have hexagonal but in 3 dimensions, icosahedral, various points of attachment. Those are the ligand interactions. The vesicle may be distorted by the amount of protein in it, but the lipids themselves would tend to be spherical. Thank you.
Thanks.
For that question. Okay, so I want to spend a good chunk of time today talking, because it's of interest to me, talking about this part, it's also the subject of the class, is attachment. What does it mean to attach? What does it mean to be weakly bound and reversible? And what does it mean to be strongly bound and irreversible? So let's start with the assumption that the virus has to bind to something on the cell. It's going to bind to a receptor. How do we know that a given receptor, for example, let's say we're early HIV scientists and we want to figure out, we think maybe we have some evidence CD4 is a receptor. How do we show that it's the receptor?
Tag them both.
Travis?
Tag them.
Tag them?
Yeah.
What would you...
Like
you're it?
No, like something that fluoresces.
Okay, fluorescent tag. So yeah, think back to the methods chapter. And so,
like phosphorus.
all right, let me make CD4 blue and GP120 green.
Okay.
How do I show that they're receptor and ligand?
And then
Inject the virus in the...
Yellow, green make blue? Wait, what did I don't even remember what I called it.
green, blue.
Yeah, that's right. I was thinking back to pre-K. So what would you do? Look at it in a confocal microscope?
No.
It wouldn't prove anything, right?
Right. So then you
could show co-localization.
Okay.
I'm sorry. So here's what Travis is seeing as he's upstairs with Marcus Davis yelling at him to get off the scope. I don't know what T cells look like. Let's say they look like that.
Okay.
And CD4 is blue. Let's assume it's bright enough so that you can see some punctate blue spots. So we're talking a single molecule, which is almost never doable. And then we're going to see, let's make this purple. I don't know what shape HIV is offhand. And then we have green. So if you have that image, you've got evidence, but you're not showing. Could be another protein right beside GP120. It could be something else you're not seeing in the cytoplasmic membrane. You probably have the start of a good doctoral thesis and perhaps a Nobel Prize if this is in the 1970s. All right.
You isolate the protein?
Isolate the protein. Okay, I've got it in a tube. I'm not speaking as mechanically as you're getting. What are you attempting to do with your methods here?
Can you just take CD4 out, completely eliminate it, and see what happens?
You mean how?
Or not CD4, the, what was the question again?
All right.
Well,
BGs are playing.
so you could.
I'm wearing a wide collar. There's a new viral outbreak we're very concerned about. We have some evidence. We know people infected with the virus are losing their helper T cells and dying. We have, I don't know what it is, but we have some suspicion that GP120 and CD4 are the ligand receptor pair that results in HIV attachment to T cells.
So
Can you,
How are we going to show it?
can you get rid of one and see what the effects are?
right.
How, what do you mean? Yeah.
Like take it out?
What does take it out mean?
Deactivate?
How
Yeah.
do you deactivate?
Knockout?
something
Gene
to it?
knockout?
Okay. Yes, you could take CD4 out of T cells in humans.
No.
No,
have to do
like
it
a little animal or something.
A
Like
cute one? How about a mouse?
Yeah.
Right, that's number 5. Is show resistance in a knockout. That was supposed to be my big closer, by the way. So there are a couple of problems. What might you imagine would be a problem with that experiment? So you get a mouse, you do all the molecular biology. It's very expensive, by the way, and you wind up with a mouse with no CD4. What happens?
And then you try to infect him, and he either does or doesn't get infected.
Right. So what's the problem with infecting a mouse with HIV?
He's gonna die
Is he?
if
Well,
he if it
you don't have
infects.
CD4, you won't die, right?
Right.
You're not going to propagate the virus without CD4.
Okay. Okay. So let's say all that happens, right? You get your mouse, you knock it out, you infect with HIV. It doesn't get HIV. It doesn't get infected with HIV. Have you shown that they bind?
No.
No.
You've even got perhaps a larger problem than that. Yes, Tracy. Alana.
Well, you can't show it, it's only in humans, HIV.
Yes, it's HIV,
Oh, yeah.
not murine immunodeficiency. So you got model problems there, right? You have to have a virus that's
So
capable of infecting mice. You have to have a model organism that's capable when you knock out of
So you
showing
have-
the mouse may be resistant to HIV anyway, probably is.
So you'd have to do it with like human.
So we can go way down that road. There are humanized mice. There are mice that are model systems with human immune systems. All that's it's number 5 because it's really hard to do.
Use things like cancer cells, since they'll be
That are perhaps overexpressing CD4, yes, perhaps. But I'm trying to, let's go to the other end of the spectrum. How do you show their receptor and ligand?
So you pay a catch?
Yeah, show them And you were headed that way. You get them in a test tube, and then what do you do?
Put them together.
And then what do you do? Try to take them apart. Okay, yeah, that's what we're going to talk about next. You can then do.
That'll be awesome.
Show they bind. Show binding is disrupted by, in this case, soluble domain. So we do have a problem here. Both of these are CD4 is a membrane protein. Soluble derivatives of the receptor, in this case CD4, that normal or by biological receptor blocks GP120 binding. So in experiments 1, 2, and 3, we can isolate the molecules. That might be difficult to do, but if you can, you do. If it's not pure, you can work in impure solutions. You can need radioactive tags or fluorescent probes. We'll talk more about this in principle in a minute. 2 and 3 are very similar. You show that binding between GP120 and CD4, perhaps in a cell, is blocked by a soluble domain of CD4. So if you had a plate of cells and infected them with virus, and then right beside them a plate of cells to which you would put soluble CD4 in the medium in great excess, perhaps that will result in fewer infected cells. That would also be good evidence. MHC2 binding to CD4 might block GP120. Both of these are what are called competition experiments. And are showing binding, measuring binding is great. Showing competition is even better. And then somebody already said number 4, which is non-susceptible cells. You can transfect another cell line. Oh, let's say Chinese hamster kidney cells, baby hamster kidney cells, BHK cells. We use them in our lab all the time. With CD4, human CD4, if those cells then get infected, that's pretty definitive. So we have a number of things you would try to do to show the ligand and receptor are the biological ligand and receptor. This is an aside, I really like to show it. This knocking things out in a mouse and showing biological effects is a relatively new phenomenon, at least for me. I was in grad school when it started happening with great frequency. And my favorite paper in this is the cannabinoid mouse knockout model. So this paper was published 21, 22 years ago, something like that, where they took the wild type. This has nothing to do with virology, by the way, but these sorts of data would be seen in a virology paper showing the same thing. Took a mouse, knocked out the cannabinoid receptor 1, 2 cannabinoid receptor. The knockout mice were perfectly normal. They showed no defects in neurology, growth, reproduction, anything. They looked like totally normal mice. And this is a little window into what your life as a grad student might be like. So what we see here is the big figure from the science paper. Science papers tend to be very short. Big figure showing the data and then explained it in later papers. If you take, and then, so cannabinoids cause pain relief, analgesia. You have there's an assay for that, the details of which are not important to it, I don't remember. That's what happens when you take a normal mouse, a wild type mouse, and give it 10 mg per kg of THC. You get analgesia. The knockout mouse, as you would expect, you don't. So cannabinoid mice don't experience analgesia from being dosed with cannabinoids. You can't see this because of the screen here, but they don't get stoned either. So this is quantitation of mice getting stoned. This assay right here is tail immersion. So if you take a mouse and you stick its tail in hot water, getting this right. Hang on a second. Yeah, this is backwards. Yeah, oh, sorry. Let me skip over this. This is normal mouse stone based on movement. So the mice that get stoned stay stiller. The knockout mouse doesn't. And then there's some great ones like piloerection. So pinpricks on the skin you get, normal mice. experience at knockout mice. Well, sorry, this isn't working out. Now I'm making mastication. Stoned mice chew more. This is quantitative munchies here. Knockout mice don't. See, the model here is just for entertainment purposes, but the point being is if you take knockouts as was proposed, or include in a system that doesn't have it as sort of a knock-in, if you would, and then could biologically show attachment and entry, you have evidence that your receptor and ligand pair are indeed correct. Oh well. So what are we talking about when we're talking binding between CD4? So here's your textbook's imagination of what CD4 looks like, which is almost completely uninformative, except it shows you it has a number of domains, and those domains fold up on their own. So from a macromolecular point of view, I thought I should throw some ribbons in there and say, binding is not, well, binding is A plus B going to AB, but at an atomic level, it's favorable interactions occurring. These are two domains of CD4. I believe they're this one and this one, but I'm not certain. This is GP120, a domain thereof. And what you're looking at is real molecules that really bind one another, and they do so by non-covalent interactions. So what you're seeing here is GP120 and CD4 fitting together. So your book says lock and key, that's the general analogy, but we're talking about hydrogen bonds, hydrophobic interactions, charge-charge interactions, and van der Waals. These molecules fit together, and it's always non-covalent. Non-covalent, so GP120 and CD4 do not react. They bond. Binding is always reversible. Second, so your textbook is quite wrong when it says binding is irreversible. And I want to take some time to talk about why that is. So, viruses, weakly bound at first, additional interactions occur, and then they're bound strongly. And irreversibly. Technically incorrect, it's the sort of thing that really bothers biochemists. Non-biochemical biologists are not troubled by this, but the difference is important. As long as you realize it, then you can do whatever you want. So here's a binding event. A plus B go to AB. The double-headed arrow in the middle indicates what?
Reversible.
Reversible, other direction. You're right, say it properly.
Equilibrium?
Yes, they're in equilibrium with one another. And we're governed by rate constants. We'll call them K on and K off. So if A and B are in equilibrium with one another, and they have to be, well, they're not covalently bound. So it could be something like this. That would indicate, sorry, this is shorthand for equilibrium lies very far in the way of the bound state. This would indicate, this is shorthand for strong interaction, and then weak interaction is the other way. Let's now imagine a more complex viral attachment event, and then back up and do the simpler events. here's the simplest imagining of the naked viral attachment event in the figure from your text. You have one virus, three receptors. So all this is saying is, I guess, GP120 is A, CD4 is B, if we're taking this to the HIV analogy. Oh, sorry, A is actually virus, so really this is, it's not one GP120 binding 3 CD4s, it's one virus binding 3 CD4s. We'll assume for a moment that they're absolute, getting too far in the weeds. We're assume that they're all the same affinity. So A and B going to AB is in equilibrium with rate constants K off, K on. I'm going to go to telling you about KD, which is the equilibrium dissociation constant, a ratio of the two. So I want to use this to draw a distinction between tight and weak binding. If the on rate is very high relative to the off rate, as I've drawn in my arrows here, we get most of the A in the equilibrium is bound to B. In other words, the majority of our population is in the AB form. Now, if it's weak binding and it's reversible, then we're going to see dissociation. So this would be virus attaching to cell and then detaching from cell and floating away. So from the virus's point of view, a non-productive interaction. But with a second receptor in the equilibrium, the bound form then can bind with another one to form AB2, or a virus with 2 receptor attachments. That actually lowers the concentration of AB. And what does Le Chatelier tell us about what will happen to the first equilibrium?
It'll shift to the right.
Shift to the right. So the more attachment points there are, the less free, the more bound, the more bound, the more bound. So that step between reversibility and irreversibility is not a chemical one. It's just a practical biological one. When you have a virus bound to multiple receptors, it's not coming off because all of them would have to dissociate for the virus to detach. Now, the real world is much more complicated because this first binding event is actually going to increase the concentration of the second receptor as far as the complex is concerned, because this first interaction is virus coming from bulk solution to surface of the cell. There are many more receptors in the surface of the cell than there are in cell plus bulk solution. So it's likely that the first binding event is the determinant one, and very quickly we go to these. But I want to put a quantitative spin on all of this, which is, what is tight binding and what is weak binding? What's irreversible? What's, for our purposes, reversible? And so to do that, I want to send you back to the horrors of general chemistry. So, what's an acid?
Something with a high
It's a proton donor.
No.
It could be a proton.
Thanks for trying.
Higher
A proton donor is one.
I'm sorry. Carlos, you changed positions. Freaks me out. Anyway, Carlos, what would you say?
Proton donor.
Yeah, something that donates a proton. There are other definitions, but we're biologists, that's good enough for us. Here is a weak acid in equilibrium with its conjugate base. So we have donor of a proton. Once it's lost its proton, it's a conjugate base. We talk about acids with pKa. So does anybody remember what a pKa is?
Isn't it like the measure of,
Well, the Ka, that's correct.
is it the measure of like the availability of that proton, like its ability to donate? Like, the
Yes,
lower the
that's,
number,
that's exactly where I'm
the
headed.
lower it is.
So, what's your name?
Brandon.
I always think he was fire department guy. Just gotta have this, isn't it? Brandon, so Brandon says Ka is the negative, pKa is the negative log of Ka. You're absolutely correct. That begs the question, what is Ka?
The acidic dissociation concepts.
Yes, the equilibrium dissociation constant of an acid. So Ka, pKa, and Ka tell us about the strength of binding of that conjugate base to its proton in the acid form. So which one of these is the stronger acid? By stronger, I mean more ready to donate its proton. More readily dissociates from its proton.
Acid number 2.
Acid number 2 is our choice. What's your reasoning?
Go lower pK value.
What does that mean? Right, that's my question. What is having a lower, so yes, you're right. What does having a lower pKa mean?
Well, that
Or
proton is
associates.
I'm sorry, what's your name?
Nicolette,
Nicolette. What did you say?
that it more readily disassociates.
More readily dissociates. Or at equilibrium, we have more
Conjugate.
in the conjugate base form than we do in the protonated weak acid form. So the lower the pKa, the more we're shifted over to, I'm blocking it. I'm doing this on the seesaw here. So HA, A minus. So the more readily donated the proton is. And so we're writing, when we talk about acid-base chemistry, we write in the reverse direction than when we talk about receptor biology, but it's the same thing. So the equilibrium dissociation constant, equilibrium dissociation constant is the ratio of the concentration of reactants to the concentration of products. So a pKa of 4 means Ka is 10 to the minus 4, or for every 10,000, sorry, I'm doing this in, oh no, for every 10,000 acids, we have 1 in the conjugate base form. 10 to the pKa of 1 would be 1 to 10. So for any given number of acid molecules, we have more in the proton form than we do in the proton form for the weaker acids. The stronger acids, the ones more readily dissociates. So I bring that up by way of introducing the idea of there being a refresher in acid-base chemistry would help with the what we're about to talk about pharmacologically speaking. So you're correct in that now we're going to call this is, I get confused sometimes here. The Ka is the equilibrium dissociation constant of an acid. Ka is a Kd. So Kd is the equilibrium dissociation constant. It is the quantitative measure we use to discuss strength of binding. So at equilibrium, the equilibrium dissociation constant, so we're talking about the ratio, the equilibrium in that direction with the products. There's no chemistry going on here. Is the ratio of free to free reactants is the wrong word, but free to bound. Note that this has units of molarity. This is molar times molar divided by molar or molar. And it's also the ratio of the rate constants, for reasons we won't go into that. Let's go into them. At equilibrium, AB forms with AB formation is K on and A and B, and AB degradation is K off and AB. So K off over K on equals AB over AB equals KB. So KD's equilibrium dissociation constant is the ratio of the rate constants, and it tells us about the strength of an interaction. And so we have a KD of 10 nanomolar versus a KD of 10 micromolar. Which is the stronger interaction? It's not rhetorical, I'm asking. Take a guess, you got a 50% chance of starting.
Top one.
The second one, micromolar. Why?
Large grain of manna.
So it's the equivalent of having a
Yeah.
pKa of, sorry, no, wrong way to go. So, let me go a different direction. This number is how much smaller than this number? Or as normal people say, 10 to the 3. So that means, given the same number of molecules in two different equilibria, one in a beaker over here, one in a beaker over here, the one with the 10 nanomolar, now you're going, except it's 1 times 10 to the minus 8. You're right, you can move the decimal around as you like with the exponents. We're going to have more in the bound form relative to this, because this is a larger, or this is a smaller number, so this fraction has a larger denominator. So what I want you doing as we step through virology and as you move on in life and talk about drugs and enzyme kinetics is being able to look at these numbers and go, oh, that's tighter than this. Oh, micromolar binding, that's pretty weak. Nanomolar, that's pretty strong. Femtomolar, well, that's practically irreversible. But it's not really irreversible, it's just femtomolar. So seeing is believing, measuring is knowing.
Wait, so
I didn't think of that.
a lower KD is higher binding, is tighter binding?
Yes. With, right, so the lower this number, the higher this number with respect to the products. So this is 1 over 10 to the 8, for 10 nanomolar KD. And this is 1 over 10 to the 5th for 10 micromolar KD. In other words, for the 10 micromolar KD equilibrium, far more is in the free form. So if we go back to our virus particle and say, GP120 binds with a 10 nanomolar affinities, the vast majority is going to be over in this form. If we say, let's go into the lab and make a mutation, GP120, that weakens binding, we can quantitatively measure that, let's say it's 10 micromolar, we've lost 1,000-fold factor in affinities by, say, changing a leucine to a serine. I don't know what it would be, but you can get quantitative, and as you get quantitative, that actually gets to the understanding. So I also want to spend a few minutes talking about how one measures KD. And I'm not exactly sure how I'll ask you this stuff on the test, but it'll probably be interpreting data that I showed you. I'm going to build a simulation here just so you get a feel for it, and then we'll go from there. So let's say we want to go into the lab. We don't have a knockout mouse. The mouse isn't going to get infected with the human virus. But we do have a purified CD4 and a purified GP120, and we can separate bound from free. That could be chromatographically, bound is bigger than small, so you can do a separation. Sorry, bound is bigger than free. You can separate free from bound. Let's just take that for granted in any number of ways you can do that. Oh, I needed that. So we want to describe, we want to do number one, show they bind. So we want to show they bind, and actually we want to measure it too. So let's say A is our GP120. So we can, and we can control the amount of GTP we put, GP120 we put in the tube. We can control the amount of CD4 we put in the tube. What we can't control is the fraction that's bound and the fraction that's free, right? It's the equilibrium that's going to control that. It's the affinity of A for B that's going to, of A for B. So we can control the total amount, which we call AT. And that's, of course, going to be equal to the concentration that's free plus the concentration that's bound. And a great leap there. So KD is equal to free, which for our purposes here is total minus bound times the concentration of B. What we're going to do in this experiment, we're going to hold the concentration of A, our GP120, constant. We're going to vary the concentration of B. For now, we'll just worry about this. Then we can multiply both sides by AB and distribute that B. We can subtract A, B, B from both sides and isolate terms. And then we can divide both sides by KD plus B and A total and express a function as this. So I'm getting to,
Why didn't you do KD times AB plus concentration of B? I don't see why you wrote it that way.
I did two things at once.
Okay.
I subtracted this term from both sides, so I would have KD B plus AB B, and then I
Gotcha.
extracted the B term. All that, the real answer to your question, Travis, is I can measure this.
Okay.
What is that?
That's your bound A.
That's the amount bound, the fraction that's bound, that fraction of A that is in the AB form. So there's an inherent assumption that you have to make sure, when you're an experimentalist, you have to make sure of, which is that B, the concentration of B is much, much greater than the concentration of A, so that we can vary the concentration of B and measure binding and determine KD. So how do you do that experimentally? Easy. Not really, actually. I spent about 3 years in a cold room isolating molecules so I could do binding assays just like this. But now I can do them in a couple of minutes, given the right situation. So I want to show you some data here. So this isn't real data, it's data they're data that are gorgeous. So. Okay, so let's go into the lab in our minds. We are going to vary our concentration of CD4 in 6 different samples. We're going to hold our concentration of GP120 constant. We're going to mix the two, allow them to come to equilibrium, and then separate bound from free. Again, there are any number of techniques that you can do this with that are unimportant. Well, they're very important, but they're not important for thinking about the mathematics of this. So here are our data. I just built this right before class. So this is some unit of concentration of B. Let's call it nanomolar of B. Then this is binding, which I'm measuring in some unit. The nice thing about this is it doesn't really matter what your units are. This is what we would refer to it as fraction maximal binding. Right? The maximum amount of binding you measure in this experiment is when all of the A is in the AB form. So that would be AB equals AT. Practically speaking, that never happens because of equilibrium. But you can get close enough to 0 that it's practically 0. You don't have to know what it is ahead of time because if you have, let's just measure bindings. Let me just rewrite this. I'm writing it in terms of concentration, but it could be in degrees of blueness if we had a colorimetric assay. Sorry, I'm running out of space. I'm going to have to erase. All I'm going to do is multiply, oops, it's AB, both sides by AT. So measuring binding in some arbitrary units, it means we have a rectangular hyperbola. There are at least 2 people in this class who have survived my biochemistry class. And you'll recognize the math exactly from Michaelis-Menten kinetics, which are really no different. You have to make a bunch of assumptions in Michaelis-Menten kinetics. But we can fit that with a rectangular hyperbola to the point where, with a simple formula, we can take experimental data and extract the constants. I just wanted to do that for you because I have a lot of fun with it. So when you go into your scientific graphing program, you guys have probably done, you must have done linear regression on Excel where you click a button that says linear regression. How does Excel determine the line? Brandon? Okay, how does it determine what the, so Brandon says it finds the best fit line by linear least squares, is that correct? But how does it get there? So it finds the line that goes to the points best. It can do that, well, I don't know if Excel can, maybe probably does that. You can do it with any number of statistical tests by finding the line that's the closest to all of the points, but that's, how does it get to that point? I'm not asking for the deep coding way of doing it here. You give Excel a bunch of lousy data from your Chem 2800 lab where you do the potassium permanganate type. I'm sorry, bringing up bad memories for some of you. You give it some data, you say, tell me the line. What is it doing?
It's finding the
How does it, okay, it's finding the best fit line. How does it do that?
It finds points that are in
Measure the
between each one.
deviation on points,
Yeah.
average it out.
But, so where do the points from which it measures the deviation come from?
Data.
You put it in the system.
Does it just find the slope of the x and y intercepts?
Well, the slope and the intercepts come from the linear least squared fit of the data. It's got to have something to determine the linear least squares and the slope and the intercepts from. Magic is not the answer. Let me try it a different way. How would you determine the best fit line given a piece of paper with these data on it?
Take a ruler.
Well, a ruler would be a bad fit, right? But let's do one where we can do
More data points?
Yeah.
Take more
might
data?
actually be able to teach you guys something useful today.
Take more data?
Okay, there's my Kim 2800 standard curve. How am I going to determine the best fit?
What's gonna include the most points?
Is that the best fit? No. Did you say sure? Well, one thing's for sure, Brandon's done with his lab report and he can go out and party on the weekend. But that's not the best fit. Let's do linear least squares. Here's the distance. Right, so we're going to square the differences. We're going to find out what they are. I'm going to find that this line may be a better or worse fit. Let's say it's worse. And I'll do that.
Right.
That looks like the third one looks a lot better. The best fit, I don't know, but it's the betterest fit. So what Excel and any sort of computer algorithm, and there are a lot, I hope as you finish your days here at KSU, you'll get into some more complex modeling, does, is it guesses.
Right.
And then it measures. And then it makes another guess. And it measures.
It doesn't a lot, really.
And it does, yeah, that's the advantage of the computational powers. It can do a lot of them and very quickly come to the best fit. And how does it determine that?
It measures the distance.
It makes a guess and it doesn't get any better. So as long as it's making guesses and getting better, it keeps guessing. When it gets, I'm sorry, I'm a biologist, right? This is how I understand computers. It gets better until it's better. And your eye is almost as good as Excel, right? You can do a best fit linear least squared just by looking at it. You can even do it. This is perfect data, right? Real world data are not like this. But let's say you want to fit these data. So we're going to do a simulation to show you what binding data look like so that when you see them, you can not only appreciate them like a fine micro-brewed craft brewed IPA, but you can understand what they mean, compare them to other things. It's the quantitative aspect I'd really like to drive home. So let's go in. All right. So I measured binding. So I varied B. I measured binding. Here are the data. They fit that model y equals ax plus b plus x. Because they do. That's what a rectangular hyperbola looks like. We've done the algebra. We know that in a simple system like a plus b goes to ab, this is the case. So what are we doing? We're guessing. So what's built over here is what's called a simulation. So what I have are the two constants my x, and we're going to calculate y based on what those constants are. So, equals A total times B divided by KD, KD plus B. Oops, not B. B is, let me just explain. So what I've done is I've gone down this column and generated a fictitious number of samples of B varying by 0.5 unit all the way to 50. So I'm covering this span. And I'm going to calculate for these two constants, what would my binding be? And so for those of you who haven't experienced the wonders of Excel yet, when you copy and paste, Excel will slide. Well, I don't want these to slide, why not? They're constants. I like to ask this on my advanced biochem final. Does the rate constant change? And the like 8% of students say, well, yes, it does. Of course it doesn't, because it's a constant, but they're constants. And so I'm going to go in, and if you didn't know before now you do. You put a dollar sign. Oops, I'm putting it in the wrong spot. Should be around J. Put a dollar sign in, that locks the Excel. So that as I copy and then paste, for every one of these cells, and by the way, this is biology, so you don't have to worry about significant figures to that. That's calculating for 0.5, that's a nanomole, or B, we would see 1.7 binding. Let's go add our simulation. Ooh, it's already there. So all I'm doing is telling Excel to put the XY coordinates I just generated from my calculation on the plot. So, did I achieve a good fit? I know I did. What should I? So, graphically, total binding. So imagine a rectangular hyperbola. Total binding. What's a good guess for total binding? Maybe 70. So I did do some other math here. What should I change? KD or AT or both?
AT.
What should it be?
Like
18? No. 80. Did it get better?
yeah.
I see why.
So change your KD now, I guess?
Change my to what?
To like 25, I don't know.
Okay, watch what happens. Everybody already knows you're wrong.
Okay.
Let's show.
I really don't care.
Being wrong is good. It's an iterative fit. You have to. You're wrong. You were wronger. The curve moved away from the data, so what should we do?
Go down.
Other direction. Let's try.
We're trying to show that.
It got better. Well, because Travis made a bad guess, I knew to go the other direction. I also know what the answer is.
There you go. Yeah. I like 68.
So, that's good enough. I just changed the constants. Can't do that.
Yeah.
Constants are constants. I didn't change the constants. I didn't know what they were. Now I do. So, for any given set, I discovered what the constants are. I didn't know. Well, I might have known. In the example I gave you, I did know what AT was, but I don't have to. I could have ground up rat brains if I was looking. Could I have known percentage and not know this number? But I either know it or I didn't. Let's say I didn't. So maybe it is. Let's say it's 70, and I know it's 70. It's not changing what I think my KD is very much at all. So I've got a simulation that goes pretty close through most of my points and pretty close to the other ones. And by the way, the difference between 5.5 and 6 is nothing. And so my total binding is somewhere around 70. And that's what it would be if I could get out to infinite B concentration. I would approach the asymptote of that. I just want to run through the data analysis inherent in determining a KD. So if that KD were 1,000-fold weaker, we'd hardly see any binding at all at these concentrations. By then, maybe my concentration's 1,000-fold. So that nanomolar versus micromolar KD is a number on a page, but it's incredibly informative. 5,500 in this fictional example is weak binding. It's almost non-existent. That doesn't mean it's not happening. A binds to B, B goes back to A. It just means a lot of it's not in the complex at any given time. Conversely, the 5.5 is tight binding. What if it were 1,000-fold stronger? It would practically be at maximal binding with a tiny fraction. We'd have to do additions much smaller in our experiment to be able to measure that binding. And that's if this were a nanomolar KD, this one was micromolar, this one was picomolar. So a receptor-ligand interaction that's picomolar might be good enough for getting viral entry based on one interaction. Nanomolar might be good enough based on getting us to that AB plus ABB to AB3. But you can get quantitative, and we will as we go through the semester. And that's your foray into binding. I was going to get into kinetics, but I won't, because I'm running out of time. So what I would just to sum up that, what I would hope you would be able to do is look at binding curves and understand something about the comparative affinity. Saying something's micromolar in KD, I know means weak binding. I don't really know what that means, but I know it's weaker than nanomolar. 1 micromolar would be weaker than 0.5 micromolar. Quantitatively less so. Or not as starkly quantitatively different as a much lower KD. Okay. So to get into the cell, what is this, attachment and entry? That was attachment. Let's talk entry. To get into the cell, viruses have to cross a membrane. That can be a direct membrane crossing event, such as phage injection of the genome into the cell itself. More commonly, particularly for animal viruses, we have endocytosis. So that endocytosis can either be we have endocytosis forming a subcellular vesicle, which is, again, clathrin-coated, clathrin-mediated, caveolin-mediated. They're very similar. Different viruses will use different ones, and we'll talk about those later in the course. But again, this protein, in this case clathrin, causes an invagination of the membrane around the event that is actually a signaling event when the virus is bound and collects multiple events that clathrin as a result of that binding that result in the invagination and endocytosis into a vesicle or liposome. Then the clathrin falls apart and you have an endosome. This can also happen at the cell surface. So in the enveloped figure, you notice there are two different types of events, which is you have, so we just reversible, irreversible. This is the relatively weak binding event followed by more relatively weak binding events, the sum total of which is a strong binding event, which we're calling irreversible, although we're now much wiser than that. You can have a fusion event at the membrane with viral entry, or you can have endocytosis, and then there has to be another fusion event, because with an enveloped virus, you have, with endocytosis, you have the endocytosing of a liposome, so you have a liposome within a liposome, and they both have to, fuse to one membrane to escape. So, do you want to, this is sort of gastroenterological framework here, so. Everywhere from your nose to your anus is actually outside of your body. For those of you taking developmental biology, look at blastulation, gastrulation. Being in a vesicle, in an endosome, is being outside. So this virus particle is outside. At this fusion event, it's inside. We have lipid bilayer meeting lipid bilayer and a fusion event. Here we're inside, but yet we're outside until the fusion event brings the outside in. And I'm very confused because I kept reading about fusion proteins in your chapter, and that means a different thing to me than it does to virologists. A fusion protein to most other molecular biologists and biochemists is two different genes fused together and then expressed as one polypeptide, so they're two fused together proteins. This chapter is talking about fusion proteins involved in fusion. Lipid bilayers will not fuse spontaneously, i.e. without those proteins. Why not?
Keeps the outside out.
Well, they're good at compartmentalizing you from the rest of the universe. Yes, that's right. But why are they good at that? I'm asking.
Well, without the proteins, nothing will interact, right?
Why not?
Cell wouldn't have recognition of anything.
The proteins are allowing lipids to bring stuff in, right?
I don't know. This is oh no, that's my cell. Sorry. That's, keep drawing my cells a second nature. So. There are 12 lipid molecules in two different bilayers. So globally they're spherical. Why don't they come together and just merge into one? Is it favorable? Why not? So lipids will spontaneously form bilayers, but they won't spontaneously fuse bilayers. Why is that? Polarity. Yeah, well, yes.
Well, they have to.
The real answer is the laws of thermodynamics, but polarity is close enough. In order to get, say, this lipid into that orientation, we have to do, or this orientation, we have to, yeah, we have to use a lot of energy, relatively speaking. We have to pay the thermodynamic cost of solubilizing these acyl chains in an aqueous environment. And so the tails of lipids spontaneously form bilayers because it's a non-polar environment. To get them from one to another, you have to move it through a polar environment to get to another perfectly stable structure. So the kinetic barrier to moving from one vesicle to another is so large as to practically never happen, unless there are proteins around to catalyze that transition. And so we have proteins in the virus itself, and in many ways they're not well understood, that catalyze fusion. I'm I'm basically out of time, but here's the example from your textbook. You have a viral coat protein from an enveloped virus. You know the little red thing there means protein. We have a trigger. That trigger may be, for example, pH shift. What would happen in a drop of pH? We'd have a lot of acidic amino acids protonated so that they would become uncharged, and that could cause a structural perturbation, exposing a hydrophobic sequence, which then buries itself into the other bilayer and can catalyze the transition of one face opening up and becoming part of another bilayer. So this lowers the kinetic barrier by fusing the two via the protein bridge. So, see you Wednesday.